Hey, what's up guys? This is Adi from ShastryNation, and today we're going to be talking about gene regulation. Alright, so now that you're back from the intro, let's go ahead and get started. So, gene regulation. So, gene regulation is basically how genes are regulated in our cells, I guess. So why do we need to regulate genes? Let's go ahead and why do we need to regulate genes? So why do we need to regulate genes? We actually need to regulate genes so that we don't overproduce on something or make things and use up resources that for things that we don't need. So we need to, regulate when we need something. So for example, like in the lac operon or something like that, which is an example that we're going to talk about, we actually, the bacteria in our body, in the stomach, I guess, they don't make lactase, or the protein that digests lactose, until we actually ingest milk or lactase, until we actually digest lactose, they don't actually make those because it's a waste of, resources. We could actually make other proteins or something like that with the same amino acids that we made with the other one. So that's why we need to, let's see, why do we need to regulate? How are we going to write this down? All right, so to conserve resources by making things when needed. Okay, so now let's go ahead and move on to where the regulation can actually occur. Okay, so where does regulation actually occur? Alright, so it occurs in the DNA. Obviously, this would probably be the most likely place to have it occur. Okay. The mRNA. Okay, and the protein. So it even happens at the protein level. Now, if you haven't watched the video on how proteins are actually made, I would go ahead and watch that. It's in my channel, or I'll try to have it linked in the description if I can remember. But I would go ahead and watch that. It's a pretty important video to actually understand everything that's going on here, and you can make better connections, things like that, if you watch it. All right. So now, from looking at this, you'll probably notice that regulation occurs at every stage of transcription and translation, or protein synthesis, and that is definitely true. We want to try to regulate and constrict everything as much as possible to what we need, rather than trying to get proteins that are doing different things to what we don't need. So that's why regulation actually occurs. All right, so let's go ahead and talk about how regulation actually occurs and where it happens, things like that. So we'll go ahead and start with DNA. So in DNA, let's go ahead and write out a DNA. Okay, so in DNA, regulation will actually be affected or done by of a gene will be done by the regulatory gene in upstream of that gene. Okay, so what am I talking about right here? If this is the gene, okay, this is the actual gene that codes for protein and everything like that. So again, transcription, translation all occurs here. Upstream of that gene, however many nucleotides up there, it just depends on that. The amount of the distance between the gene and the regulatory gene is just dependent on the gene itself. But so let's say this section over here is the regulatory gene. And so what happens here is that it'll actually the regulatory gene will actually make a regulatory protein, when it's actually coded for, it'll make a regulatory protein. It'll make a regulatory protein that will actually act as a operator or a operon, you know, or something like that to actually regulate this gene. So it'll go, this regulatory protein, which I will do in pink, will go here and actually bind here to stop, you know, RNA polymerase from coming over here and doing its thing. All right, so that's what happens at the regulation. So I guess some examples of regulatory genes, regulatory proteins, sorry. So some examples would be like transcription factors, yeah, so transcription factors, I guess operators. So operators are basically things like you know, things that will basically say yes or no. Okay, yes, we do need to, you know, express this gene, or no, we don't need to express this gene. That's basically what it'll do, right? And then the repressor would also be an example. So basically what it'll do, it'll go and bind to the DNA molecule and restrict RNA polymerase from actually going through and doing its thing. So transcription factors we'll actually talk about when we talk about eukaryotic gene control, because that's where transcription factors are actually used. Now, you know, you might be asking, why not we just talk about eukaryotic gene control in the first place? Well, eukaryotic gene control is a little bit more, I guess, complicated than prokaryotic gene regulation. It's a lot more simpler. It's a lot simpler, and so we can actually explain it easier. Alright, so our first, I guess our only example in this video, but there are many examples of this, but the first example we're going to talk about, let's go ahead and draw like a divider. Alright, so the first example we're going to talk about is the lac operon. Alright, so lac operon. Now, I'm going to draw a gene molecule, DNA molecule. Alright, so it's like that. Alright, and we have our, base pairs right here. Alright, so we have our base pairs right there, okay? So now, what will actually happen is that we have the promoter region right here. Well, in this particular gene, we have the promoter region right there. We have the repressor right about here. Now I'm just trying to set it up, and then we actually have our genes. So I'll make that in blue. So gene 1, gene 2, and gene 3. All right. So the promoter, repressor, and gene, and the 3 genes. Okay. So the promoter, what happens at the promoter is that RNA this is like the if you remember from transcription and translation, the promoter region is the place where the 10 or 20 base pairs where RNA polymerase can actually go and attach to the DNA molecule to start coding for a gene, you know, and these the promoter region is usually made of RNA molecules, and it's probably like the same, you know, bases, like A all the way through, so that it can actually go and attach to the DNA molecule, so it'll code for the whole gene. All right. But what happens is, okay, so we'll have like the, this will be a very messy drawing, but we'll have like, you know, RNA polymerase, that's going to be the yellow thing there. It's going to go here, but it's going to be stopped right here because of the repressor. So, you know, how do we take the repressor out? Now, this repressor is actually a protein, so it'll go all the way here, you know, it'll be thinking like, oh, I need to go and code for this, but it'll be stopped by this, and no transcription or transcription, translation, or anything of the gene will occur until this repressor is taken out. Now, in the lac operon, what will actually happen is it will have you know, let's say you eat lactose or you drink milk or something like that. So we'll have that in, I guess we can do like a white. Okay. So let's say you drink, you know, lactose, or you eat or drink something, some dairy product. So you eat like ice cream or something like that. And all these things come. Okay. So these are all, lactose molecules. Let's go ahead and write lactose. So what will actually happen is that this lactose will actually come here and bond to this repressor. The repressor, so basically this repressor is actually a protein, and so when the lactose actually comes and bonds to the protein will actually change its shape, and it'll get deformed, and it'll lose its bond on the DNA molecule. So it's going to be hard trying to, oh, that's way too big. Alright, it's going to be kind of difficult to actually try to erase this, but we'll try our best and recreate everything else. Alright, so let's say, you know, this bonds to it, okay, and the repressor molecule will actually go here, okay. So the repressor goes from here, it's still an eraser, okay. So the repressor goes from here to here, and you'll have the operon here too. The lactose molecule here bonded to it, and you'll have the rest of the DNA molecule if I can reconstruct that one. Alright, there you are, yeah, okay, so that's it. Alright, now we actually have the DNA molecule. Now what actually happens is that since this repressor is gone, RNA polymerase can actually go through and code the gene and make an RNA molecule. So this will actually make mRNA. Alright, so it'll actually make mRNA and it'll code for the gene and everything. Everything's going to be happy, and so this will go into the ribosome and make this will make like this protein, lactase. This will go and digest all of these things, okay? And it'll take all of these out. So let's go ahead and erase all of these, because these are all going to be digested by this enzyme, okay? Or this protein. And so then what will happen is that this protein will then be taken apart, and the amino acids will actually be, stored for more use. This thing will be taken apart. This thing will go back. The repressor will go back to its original shape, okay, and it'll go back here and bond to this. So then RNA polymerase can't actually code for the gene because it'll be stopped. So that's basically how lac operon works. Now, this would be called positive. Lac operon is a positive control. All right, so it's positive control. Positive. So it's positive control. So positive control meaning, when it'll actually repress the gene all the time, and when the thing that needs to be digested or something like that comes, this certain molecule that's needed to actually unrepress the gene and to have it be coded for comes, it'll actually bond to the repressor and take it out, like we see in the lac operon example with this repressor. Now, the opposite of the positive control is the negative control. So negative control is like the opposite of this. The gene will actually always be coded for. Okay, so you'll always have something being made. So always have something being made. So you'll have like a protein being made all the time, but when there's too much of the protein or something like that, it'll come and bond to the repressor to stop it. So this would be, I guess an example of this, it's not directly an example of the gene thing, but it's like a feedback loop. So basically, what'll happen is that, this repressor will always be over here. So if we took this repressor out like that, okay, this repressor will always be over here turning it on. Okay, so RNA polymerase, will actually be able to code for the gene all the time. Now, what will happen, when we have too many proteins or whatever it's coding for, when we have too many, it'll actually bond to this. The protein will bond to this, and it'll go back here and stop it. So when there's too much, it'll stop it. And that's what negative control is. When there's too many, it'll stop it. And in positive control, when there's too less, when the digestive thing, so something outside comes in, it'll actually start the transcription of something. So that's basically negative and positive control. All right. So that's the DNA side of things. Now let's go ahead and talk about the mRNA side of things. So mRNA. All right. So in the mRNA, actually, what happens is that you'll have the control, the control of the mRNA molecule will be determined by spliceosomes or other things, that are cut out. So an example of things that are cut out are introns. So there's these things called introns, and then there's exons. So as I've already mentioned before, introns will actually be cut out from the mRNA molecule. So if we had like a mRNA molecule, it's one-stranded, so we'll have like one like that. Okay, we'll have exons. Exons will be kept and introns will be cut out. Yeah, introns will be cut out. It's opposite. It doesn't make sense. So but I guess that could be a way you can memorize it. It just doesn't make sense. You would think exons would be cut out since there's X, on it. But basically what'll happen is that you'll have spliceosomes, so that'll come by and actually take out some of the parts of the mRNA code. And so then these things will glue back together, so we'll have like a smaller molecule if we actually did it correctly. Now we'll have like a molecule that big, and this will actually go and code for the protein. Now, the spliceosomes, things like that, are also coded by, transcription factors, things like that. Sorry, not transcription factors, but regulatory proteins that'll actually come by and cut out certain parts of the mRNA molecule and make things like that. All right. So then, after mRNA, the last step of regulation would be in the protein. Okay, so the protein would be the last and final stage. Oh, one thing before we go on, mRNA molecules. So mRNA molecules can last, I think, up to in a bacteria, it's like a couple of hours. And then in us, it's like a day or something like that. But, mRNA molecules can actually be broken up and, taken out quicker based on how, when we need them, or, how crucial the exactness or the preciseness of what the mRNA is being coded for. It'll actually be taken out earlier or later, depending on when we actually need it. Okay, so it'll be, the timing is also a major factor. Or how long it lasts. Is it better? How long it can code is also an important factor. All right, so then proteins can actually be, proteins are going to be mainly regulated from the other the other stages, I guess, from the DNA. DNA would be the major regulation where it all occurs, and then mRNA, it's a little bit of regulation. Protein would probably be the least amount of regulation, because at this point, the protein's already being made, and you can't really change much of the protein. But what can happen is, you'll still have regulation that occurs, by time. So just like the mRNA molecule, it can actually, the protein's life cycle would actually be decreased based on the cell's necessity to make new proteins that better fit its needs. So basically, like timing, again with proteins, is how proteins can actually be regulated. So timing is how these are regulated. Alright, so proteins, that's how they're regulated. Now, let's go ahead and, if we can find some space. Oh, there's a lot of space here. Alright, cool. So, now let's go ahead and talk about eukaryotic gene control. Eukaryotic gene control. Okay, so eukaryotic gene control, it's basically the same thing, but there's like a different, there's some more moving parts in it. Alright, so before we talked about transcription factors. So transcription factors are basically extra molecules that will actually bind to the DNA molecule, and it'll activate, the gene. So it's extra proteins bind to the DNA. Alright, so let's go ahead and draw a DNA molecule. Alright, so we have one like this. Okay, and then the other strand. Alright, and we have the things right here, like that. Okay, base pairs. Alright, now what will actually happen is, it's actually easier to explain with one strand, because you'll have like a bending over and things like that. Okay, so basically what will happen is you'll have the operator, you'll have the all of that type of stuff, but here, what will happen is the transcription factors will actually come here and bond to this area. Oh, let me get a different color. Okay, so transcription factors will actually come here, and the DNA molecule will have to bend. Okay, so in order to explain the bending, we'll have to use one strand. Okay, so we have a strand like this. Okay, we have transcription factors right here, and here. Okay, we have RNA polymerase right here, so RNA polymerase right there, and we have the actual gene right here. So we have the gene right here. All right. Now, what will actually happen is that the RNA polymerase will actually it can't be activated, okay, until these transcription factors actually bond to this side of the RNA polymerase. So how does this happen? All right. So we actually have this little formation like this. So we have this, okay, and this would be the DNA molecule. Okay. So let me write it in the same color so it makes sense. That would be a good idea. Okay, so we have this DNA molecule that's like this, and it'll go like this, and we have RNA polymerase right here. We have the transcription factors right here. Okay, and once this transcription factor bonds to the once these transcription factors, we have some over there too. But once these transcription factors bond to RNA polymerase, we can actually send it off and make a mRNA molecule. Okay, so that's basically how genes are regulated in the cell. I know this video has been pretty long. There's a lot of stuff you need to know about gene regulation, and they will ask you in the AP test about all of these types of things. Alright, so gene regulation, main things you need to know, I guess, are what gene regulation actually is, okay, why it happens, okay, where it happens, and what are the most common places it happens. Alright, what is a regulatory gene, what are regulatory proteins, some examples of regulatory proteins, what is negative control, positive control. You'll definitely need to know the lac operon, how that works, and what type of control that is, how genes are regulated at the mRNA level and at the protein level, and basically that's about it. But also you'll need to know a little bit about how eukaryotic cells will actually regulate genes. Okay, and so basically you'll just have extra things there, and it'll fold over, and that's how that works. And you also, before I forget, you also need to know what transcription factors are. So there's a lot of information on this. You can, again, you can rewatch the video if you need anything, but I think if you learn all of this, you should be okay for gene regulation. And I guess I'll leave you guys at that. If you guys like these videos, go ahead and hit the subscribe button. That'd be awesome. And if you like the video, go ahead and hit the like button. That'll help me out and decide, what type of videos you guys like, because I like making these. These are pretty awesome. And I make these every Monday. I have, I think, 13 or 14 videos out about science as of now. I also do gaming videos and other things like that. And if this is the first video you're watching, go ahead and hit the subscribe button, because I make these every Monday, like I said before. And I will leave you at that. I guess I'll see you guys on the next one.